Hi, this is Dr. Jabali, and in this video, we're going to focus on topic 7.3, which is all about translation. So we went ahead and looked at the past IB exam papers looking for long answers, just focusing on this section. So when we're working on this, it's very important that you look at the word describe. That means the first word, which is the command. And I'll tell students that scribe is not just about words, but you actually can draw. And from drawing and annotating, you can get points. Another important thing is look at the number here. Usually if it's 1 or 2, that doesn't mean you go general. That means you go very direct to the point, but very specific. And if it's like 5 to 8, that's where you can go general and specific. That means write everything from the most common sense to the most detailed sense. So with that in mind, let's start with this one. So it says, describe the roles of mRNA, tRNA, and ribosome in translation. Now, if this is like a 3 points, then I'll go straight ahead and just mention what RNA, tRNA, and ribosome do, right? But if this was like a 5 to 8 points, then I'll go and talk about the entire process of translation. So first, let's talk about what they do. So for example, DNA, you can mention about the codon, right? And you can talk about how there is a start codon as well as a stop codon. You talk about how it gets the genetic info from the DNA, right? And then the tRNA, you can say the tRNA is the one that is bringing the amino acids to the ribosome. You can also talk about its structure, which is basically has the anticodon. And you can say the anticodon matches with the codon. And you can say that one anticodon goes for one amino acid. And of course, you got to talk about the structure of the tRNA if you want. You can always draw it. You never know, you might get the points for drawing a tRNA. And it says the attached amino acid on the 3'. And then you have the ribosome. You can talk about the A, the P, and the E, what happens in each one of them. And you can talk about how the ribosome is basically reading from a 5 to 3' the mRNA. And this way it's building what? It's building the protein. So these are just a couple of things that you can mention as the process go. So with that, let's look at the mark scheme. First, it says mRNA is with genetic code or the codons. Second, tRNA is with the anticodon. Good. Third, tRNA is with amino acid attached. Yes. Ribosomes with the 2 subunits. Okay, this isn't mentioned, but of course, talk about the structure of the ribosome. Why not? The large and the small subunit. mRNA is held by the ribosome. You have a start codon. 2 tRNA molecules attached to the mRNA on the ribosome. This is basically talking about the process translation. The peptide bond between the 2 amino acids on the tRNA. The peptide is formed. Continues until a stop codon is reached. So basically this right here is just talking about translation, this whole part. But again, if you look at this, we got most of the points. So before I go to the next question, just look here. How many points have we got? We did mention 1, 2, 3, 4, 5, 6. So here already gets 6 points. And of course, now I know this extra information I can put. So depending on what the point will give you, if it's 6 points, you got the 6 points. If it's 7, now's the time to talk more about basically building a protein. So basically, I think they mentioned that ribosome basically builds a protein, so that would be 7 points right here. So make sure you look at the points, because the points right here will make a huge difference and tell you how much you need to put. So the next question says, outline the structure of tRNA. Now the structure of the tRNA looks like this. So why not draw it? You can get points for drawing if you cannot explain. You know that the 3' has a bigger edge than the 5'. And then you could put an arrow saying that here is where the amino acid is attached. Can you draw an outline? Yes, you can draw an outline. Then you can say that there's 3 loops. And you can say here, you can label it as the anticodon. And then you can say in these loops, you have base, like attaching to base, right? A, let's say C with G. So in these loops, the base are paired, right? And that means with A with U and G with C. We can also talk about how the anticodons will match with the codon, because that's the structure of the RNA. You can also make it as it's one chain that is folding of RNA nucleotides. That's the most obvious things, but you have to put it. It's also a clover. It looks like a clover leaf. Anything else that I can think here? Yeah, the anticodons, for example, let's say AUC, these ones, the base of the anticodon don't pair up. And what I mean by that, if I put it up here, let's say make it bigger, it's like A is bind with U and C is bind with G. But when it comes to anticodon, let's say AUC, you don't see them pair up with anything. So these are basically some of the ideas about the structure. Now let's look what they have here. Accept any points above if clearly explaining using a suitable labeled diagram. So I did show the diagram. tRNA is corresponding to one chain of nucleotides. We have that. tRNA has a position and site attached to the amino acid. See, but it says you reject tRNA containing amino acid. So remember, amino acid is not part of the tRNA. It's just that the tRNA carries it. At the third terminal, that's where I put it. Good. So what I mean by the third terminal, that's basically I'm saying the amino acid attaches to the 3' right here. The tRNA has an anticodon. The anticodon has 3 bases which are not base paired as I showed you. tRNA has double-stranded sections formed by base pairing. As you can see, there's a double-stranded sections. Double-stranded sections can be helical. This I did not put. tRNA has 3 loops. And then it has a distinctive 3-dimensional clover leaf. I didn't mention this part. So looking from this anymore? No, yes. So here, what are the points that I got? I mentioned that it's made of one chain. I have mentioned that the amino acid is attached to it at the end. I did specifically say where exactly it is. It has an anticodon. I said the anticodon does not, the 3 anticodon bases do not pair. Let's see, what else did I said? I mentioned that it has 3 loops. And I mentioned it as a cover leaf. So you can see that I got most of the points. For sure, when IB mark scheme comes in, they won't ask from A all the way to I. They usually put more mark schemes and you have to target. So here, for example, we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So it has 9 mark schemes. So usually this probably would have been a 6 to a 7 points. So I have 1, 2, 3, 4, 5, 6, 7. So that means I have target. But does that mean do I stop there? No. Now I know more information. So what I do, I remember to talk about the double-strand sections, about how they are helical. And if I do this, then I get the rest of the points. So looking at old mark schemes kind of broadens your mind of the kind of things IB is looking for. So moving on to the next question, says describe the genetic code. The genetic code, it's that chart that has like the codons in here with a certain amino acid, right? And you can have so many codons for that one amino acid. So when we say about the code, we are talking about the codons. Now, how many are there? 64. How many amino acids we have? 20. We all can say that one codon represents one amino acid, but one amino acid can have between 1 codon to 6 codon. And when we say that, we call this the gene code is degenerate. That's what it means. Degenerate means that you can have more than 1 codon for that one amino acid. We also can say that the genetic code is universal, which that means all species understand this. And they understand the codons, right? Because they have the A, T, C, G, and U. So they can understand that. What else about genetic code we can say? We can talk, if you want, later on about the anticodon. We can say the anticodon, there's about 64 of them. We also can talk about the start codon, which is AUG, which is MET. And we can talk about having 3 stop codons, which these are the nonsense. That means they don't have any kind of protein. So I have mentioned everything I can think about genetic code. Let's look at the process. Ooh, the most important thing. It's made of base triplets. I have not mentioned that the mRNA is made of codon, which has 3 nucleotides in there. The most obvious thing, and sometimes you forget that, putting the most obvious. Next, it's called codons. There are 64 different codons. Each codon for an additional amino acid to grow in peptide chain, just basically saying each codon of the mRNA will represent an amino acid. The genetic code is degenerate, as I mentioned, meaning more than 1 codon can be coded for a particular amino acid. That's what I was saying, that one amino acid can have 1 codon or it can have 6 or in between them. The genetic code is universal, and that means it's the same almost to all organisms. AUG is the start codon, and we have some nonsense codon that basically the end of translation, which if you say stop codon, you're still going to get the points because it's considered as the nonsense. So from here, before I go to the next one, we can say that we got 1, 2, and then 3 and 4 and 5 and 6 and 7 and 8 points. So you definitely got to hit all the points necessarily. But now I can also add the most common thing here is this one as well as this one. So now I have a much stronger base for the next test. That's why it's I'm going to repeat this. It's very important that you get yourself exposed to the mark schemes. Next, we have here explain how polypeptide chain is synthesized in eukaryotic cells and explain how polypeptides are produced by the process of translation. Now, even though these questions are worded differently, they are both asking exactly the same thing. Now, the only difference is, if you look here, polypeptide chains synthesize, polypeptides are produced. So the word synthesize is obviously the word produce. And however, here it's saying the eukaryotic cells, and here it just says the process of translation, which means in this question, they have not mentioned the word translation, and they want you to figure out. So if you put this in the mark scheme, you get points. But if you put on this mark scheme, it gets no point. Why? Because they already said the word translation. But we're going to compare the two mark scheme, and you're going to see how much they are so in common because they're asking about the same thing. Explain the entire process of translation. Knowing that the word polypeptide here means protein. So how do you make proteins? So here we have both mark schemes. What I'm going to do is go through the two of them, and then I'm going to try to show you how you can combine these two together. So I'm going to look at the left side here, and I'm going to read these mark schemes on this side and then see if there's any matches on the right side. So the first one says translation occur in ribosomes. If we look at here, we probably don't have anything that says translation. Why is that? And it's obvious because we already had the word translation in the question. So on this one, we don't have another one. This one, we say tRNA activating enzyme attach amino acid to tRNAs. Do we have here? Yes, we do. So this one I can highlight here, and that goes with the tRNA activating enzymes, synthase attach specific amino acid to this. So you see, there's already one thing common. They talk about the enzyme attaching the amino acid to the tRNA. Then on the next one, C, small and large ribosome units assemble on mRNA, or translation polypeptide synthesis starts at start codon. Do we have anything that has start codon? Yes, we have the start codon here, which is AUG. Do we have anything about the ribosome attaching? Nope, we don't really have the beginning attaching large and small ones. We don't have it, so it doesn't touch here. But we do have this part right here, the part of the start codon, although this one is more specific by giving the example. Next, D. Each tRNA arriving in the ribosome binds to the A site. Anywhere about arriving at the A site? We don't usually have that here, but we do have something like here showing the tRNA for the next codon binds at the A site, so I would say that also considered as a point. Next, we have E here, anticodon binds of the tRNA binds to the codon mRNA. Yes, we do have that. We have here anticodon binding with the anticodon binds to the mRNA. According to the complementary base pairing A with U and G with C, do we have that? Yes, we do have this here mentioned, complementary base pairing between codon and anticodon, but we don't have anything about A, U, and C. But as long as it says complementary base pairing, they are both the same. Then the next question on G here, it says ribosomes move along the mRNA. Do we have ribosomes? Yep, we do have here ribosome moves along the mRNA. So we can highlight this one too with this one. But look at this one on the K. And the K, they does have here more in detail saying 5 to 3 directions, so you can add them. Then we have H. tRNA shifts from A to P site, and then from P to E site. Do we have that? No, here it says peptide bonds are forming. It doesn't say about the shifting, so we cannot say that one. Here, the peptide bond between amino acids is between the A and the P sides. Yes, we do. We have this, which is matching with this. Peptide bond forms between amino acids from P and A site. And then here we can see tRNA releases from the ribosome on the E site. Yes, we do have this one too, which is here, tRNA release from the E site. And then it says the cycle repeats with tRNA and process recycle repeats along the polypeptide until stop codon is reached. So this is as it grows as it brings more amino acid, but it's the same thing, but until the stop codon is reached. So you can see it's in two words, but in two sections, but it's still saying about how it goes all the way to the stop codon. Then here it says release the polypeptide and the RNA, they assemble the ribosome complex at stop codon, which we have these components at the sample polypeptide leaves the ribosome. So you can see how much they're so in common, right? All this blue, it's two mark schemes matching. However, there's a couple more differences. Now, let's look at the difference. This one says translation occur in ribosomes. Here it doesn't have it because it's in the question, but it does say mRNA is translated, which you don't have the first one. Here it says small and large subunits resemble mRNA. It doesn't have here. It also shows here tRNA shifts from A to P site, from P to E site. It doesn't mention. But it does mention about the anticodon that's found on the tRNA, and tRNA carrying the first amino acid, which is methionine, binding to the P site on the large subunit, it doesn't mention. So looking from this, you can see that you can join the blue together, right? That means the two sentences, make the two sentences into one. And that means you are joining two mark schemes into one. And then after you join all the blue together as one, then you have the yellow ones. You can add the yellow ones, and then you can put them in order. So this is what I usually would tell students. Take these two mark schemes and combine them. When you combine them, you will have the strongest mark scheme. And when you know this mark scheme by heart, no matter what's the question related to translation or what we say protein synthesis, then you're never going to get points off because you know a very strong mark scheme. And I guarantee you that mark scheme can be answered in the next 5 years of IB. So with this, I will take this part here and I will combine them. An example would be like, for example, the tRNA activating enzyme attached to amino acid. And here it says the same thing. tRNA activating enzyme basically attached to the specific amino acid to the tRNA. So they're the same. So I just combine as a one sentence. That's what I say. However, I can say there's an anticodon found on the tRNA. That's what I add more. And then here I can say tRNA carrying the first amino acid, which binds to the P site. And then from there I can say the tRNA shifts from the A to P and then P to E. So that's the way you add in these two mark schemes. I guarantee you, right now, pause the video and join the blue together and then join the yellow with it as an additional. And I guarantee you will have the strongest mark scheme for any question related to polypeptide, and you're not going to miss out. Next, it says outline how translation depends on complementary base pairing. When we say complementary base pairing, we're talking about the A, U, C, and G. If you notice, I did not talk about T. Why? Because translation is about RNA going to amino acid. Right? So here we have the codon that is coming from mRNA, and it's going to attach to the anticodon coming from the tRNA. So let's say the codon for the mRNA is AUG, the tRNA will be UAC. That's what complementary base pairing is. Now, important that every codon will eventually have its own amino acid. Ironically, the same thing, the anticodon, both of them will talk about the same amino acid. So this is what it means by complementary base pairing in translation. Let's see what we have here. Translation converts sequence of mRNA or codons to amino acids, polypeptide chain. So you can see the whole process was just changing it to an amino acid. Right here, mentioned RNA took the amino acid. Nucleotides are based with the T-prime is paired with the complementary of the codons, nucleotides. So basically, we're just pairing the nucleotides of the tRNA with the nucleotides of mRNA, and that's just basically talking about who? Talking about the codons and anticodons, so you still get points. And then I talk about the base pairings of A with U and G with C, so you get the third point there. and specific amino acids are attached to specific tRNA, like I told you. So in this process, if you mention the anticodon of the tRNA match the codon of the mRNA, you're already getting the points. Now you can talk about the anticodon mean 3 nucleotides matching with the 3 nucleotides of the mRNA, it matches again. And then of course you can say the tRNA, one tRNA goes for that one specific amino acid, and you get all the points in here. And of course, talk what translation means, right? Because you're talking about define. Translation is converting the codons of mRNA to amino acid. And that's how you get these full points. And of course, last, why not saying here, mRNA and tRNA, where they're coming from. Codons on mRNA, anticodons in tRNA. And here means if you just mention this and not mention this, you're not going to get the points. For you to get the points of the E, you have to mention both. So this is a very genetic strong mark scheme if we're just talking about translation with complementary base pairing. Now the next question says, outline the roles of the different binding sites of the tRNA on the ribosome during translation. So we are talking about who? The binding sites. And who? In the ribosome. So it's like we have here the ribosome, the large piece and the small piece. And in this we have 3 sections, right? We have the A, the P, and the E. So we're talking about how, right here, the tRNA, how it works. Usually it will arrive at the A, and then it will shift to the P, and then it shift to the E, and then it leaves. But it would arrive with an amino acid, and it would leave without amino acid. So here I will label the amino acid, and here I will say no amino acid. Then the next thing I will do is put step by step. So I can draw, because even though it says outline here, I can draw the steps. So first I would mention that there is 3 stages. You got the A, P, and E, and that's where the tRNA right here will arrive. Oops, the tRNA will arrive and lands at. So the next thing I will talk about is I can show in the drawing, and I'm going to do here a drawing that you can draw this and you get your points. So I will say the first step here, and I would put the word E, P, and A. And I would draw this just to make it clear and say these are found in the large subunit, the most common thing of the ribosome. You need to mention that. Then I would mention number 2, since you have the E, the P, A already said, you can say right here, and I would label my drawing here saying that this is the amino acid, and of course I'll write the full word, and here I will have, let's say, UAC, and I will label saying that this is the anticodon, and I will label that this is the tRNA, and here you're gonna say that it arrives at the A site. So then number 3, I can say here I have E, P, and A. I have arrived with the amino acid. Then you can say a new one is coming. So this caused the shift. Number 4, you will have the tRNA with its amino acid on P, and you have a second one coming in on the A. And then label this and say a peptide is formed between the amino acid of P and the amino acid of A. And then number 5, I can say that we have E, and then the tRNA has moved, right? And this one is moving here, this moving there. So that means you have this one with the 2 amino acids attached, right? And the A here, I can say the new tRNA is coming with amino acid, and this one is about to leave. So I can say here, tRNA leaves without the amino acid. Then number 6, again, the E is empty, and we have the P with the 2 amino acids, and we have the A with the thing, and we join the bonds, and then I can say that it keeps repeating until the stop codon. So this is just showing the step by step. You can do this and you get the full points. So looking at the mark scheme we have here, you can see AP binding sites are on the large subunit. Then we have the initiation translation that starts with the binding at the MET. So I can mention about the start codon and how that will bring the first tRNA. Like me mentioned that the first tRNA brings the amino acid known as MET. I can get points. Then here's large subunits binds with tRNA in the P site. That's the first one. And let's say it's the A binding site holds the tRNA with the next amino acid. This can be on the drawing that I showed you. The peptide bond is formed between the A and the P site. Then you eventually will have peptide is transferred to the tRNA on the A site, as I showed on the drawing. And then the tRNA on the A site then moves to the P site, or the P binding site holds the tRNA attached to the growing peptide, all in the drawings. And then the E binding site is where the tRNA exits and leaves the ribosome. And you can see here that it says the diagrams is accepted, annotated diagrams. So the drawing that I show with the APE, you can just draw it and show how the tRNA is moving from the A to the P and to the E. If you're showing with the arrows and you have the tRNA drawn here with the amino acid and everything labeled, you can get these points. You don't have to actually write these words, but the drawings with a couple words written next to them, you can get full points for explaining the whole process of how tRNA is moving through the APE of the large subunit. So moving to the next question here, says distinguish between transcription and translation. The word distinguish means what are the difference between them. And the word compare, contrast, that means what can we talk about the similar as well as the difference, right, between transcription and translation. So one thing is we can here is put the transcription. It's define them, right? Translation. One thing we can talk is the transcription is going from DNA to make mRNA. And translation is going from mRNA to make, let's say, protein. The next thing is that they both, what they do, they both use nucleotides, right? But when we talk about nucleotides, make sure you're talking about the A, U, C, and G. The next thing is transcription is happening in the nucleus. This one is happening in the ribosome, or you can say the cytoplasm. The next thing, the transcription has to do with transcribing the antisense, while translation has to do with converting the codons into amino acids. And the translation, they have tRNA, and these guys, they don't have the tRNA whatsoever. What else? Transcription, translation that we can think about these guys. They do have nucleotides. They do have the A, U, C, and G. One is a nucleus, one is cytoplasm. Just trying to think about all the possible things about transcription. This has to do with mRNA editing. So there's splicing here, right? There's the splicing of the introns, right? And here you don't have any kind of splicing. So there's no splicing going on of the introns. What about also transcription? DNA to mRNA. I'm just thinking here. Oh, this one uses the RNA polymerase, right? The enzyme. This one, we're not using the enzyme RNA polymerase. We're actually using a ribosome. We are using a tRNA, and we are using the mRNA. So this is the process that we use. Anything else? I think this is pretty much, although now the mark scheme will get me exposed and I will learn more about it. So let's see. You have the two mark schemes here. So yes, we had the first one, DNA is transcribed, and DNA is transcribed, mRNA is translated, and its end must be. And we have here both of them. So look, even both of them have transcribe and translate. Transcription produces RNA and the translation produces a protein. Do we have that? Yes, RNA. So they both are saying what they produce. We also have RNA polymerase is used in transcription, ribosomes in translation. Yes, we have that too here. See how much they have in common, even though you can see that the command word is kind of different, but they're putting in common. Let's continue here with the yellow. Transcription is in the nucleus, translation is cytoplasm. Yep, we have that too here. And tRNA is needed for translation, but not transcription. Oh, we have that too here. So here all the yellows is basically, it's the same. So you can see that they're not going to add more. So this is a very strong mark scheme. However, on the bottom part, you can see that they have common. They require ATP and they require a 5 to 3 directions. These two right here is what we considered as the similar part. So this is something else that you got to keep in mind. The yellow part is the difference. So you cannot answer full points getting compare contrast unless you also put the similar. And it makes sense. Both of them are going from a 503 and both of them need ATP to do the action. So now that I know an extra information about this, now I'm very strong on this. Now here, in the bottom, we also have nucleotides, link transcription, amino acid translation, or phosphate sugars bonds. Now look at this bottom here. Nucleotides, link in transcription, and amino acid in translation, or sugar phosphate bonds in transcription, peptide bonds. So here they're saying the different bonds, right? You can use the phosphate bonds for DNA, or coming, of course, from the transcription to RNA, or you can have the peptide bonds in the amino acids. So, or we can say nucleotides for transcription and amino acid for translation. So these are just an additional information they can add. So you can combine. You can see that the yellows are the same, but instead of just having the yellow, why not add the blue and the green into this very strong genetic mark scheme, and then you will never have a chance of losing those points. Now here says outline the role of the A site in the ribosomal translation. Now, again, this probably is 1 point because you see the mark scheme only has 1 point. This you can also draw it. So it's just basically the binding or the entry of the tRNA carrying the amino acid. Just mention that. The tRNA is entering or arriving into the ribosome, and that's pretty much it. Next, outline the role of tRNA activating enzymes in translation. So here they're talking about the enzyme. What enzyme? It is an enzyme, right, that binds tRNA to the amino acid. Now, for him to do this, he needs what? He needs ATP. So what does this enzyme do? This enzyme usually has an active site, right? Is active site for the amino acid. So he attaches the amino acid, and then he attaches the ATP, and then he brings the tRNA and attaches here. And with the energy of the ATP, right, it connects the amino acid to the tRNA. So that's what the enzyme does. It just combines the tRNA with the amino acid using ATP. Let's see what we have here. ATP provides energy for the amino acid attachment. We got that point. They attach specific amino acid to the 3N. Oh, I didn't go more details, but we know that it's on 3' of the tRNA. This is where the amino acid. So I haven't mentioned that. So now I know this information, but I didn't mention. But now guess what? Now I will make sure to mention. And they do this repeatedly. They attach amino acid to all the tRNA molecules that have an anticoning corresponding to the amino acid. So, one more information I can mention here. I did mention that tRNA is binding to the amino acid. I did mention the ATP is used. But now I can add the fact that I can say exactly where is it doing, as on a 3' end. And I can say there's a specific anticodon for a specific amino acid. So these are the extra information that I have learned by looking at this mark scheme. So when it comes to the test, I can build that up. Now, here it says explain the 4 levels of the protein structures, indicating the significance of each level. Now we have the primary level, the secondary, the tertiary, and the quaternary. All of these we can talk about relating. So this is about the bonds. So you have amino acids, and you can see that the bond here, it is the peptide bond. So this is just one chain of amino acids, and this sequence usually gives the structure. Secondary, this you already start to see more folding going on. Now this folding happens because of what? Because the bonds that happens here. Oops, let's say here. What is these bonds? These are hydrogen bonds, and these are happening between who? The amino group and the carboxyl group of who? Of the amino acids. Then the tertiary, you have both. You have the peptide, you have the hydrogen bond, right? But besides that, you also have the R groups combining with each other. So that means you might have covalent bond, such as disulfide bond. You have ionic bond. You have hydrophobic bond. These are additional bonds that are happening. Now, going back to the secondary, there's two examples. There's the spiral one, which is the alpha helix, and you need to know this. And there's the zigzag one, which is the beta sheet. You have to remember these two examples for secondary. Now, an example for tertiary is the enzyme. Okay, this is a perfect example of tertiary. Quaternary will be more than 2 chains, right, of polypeptides. And they usually have a prosthetic prosthetic group here, which can be a heme, it can be an iron, and this chain is usually an example of it will be the hemoglobin. Now, the 4 from quaternary usually has a mixture of everything, the first, the secondary, tertiary. Now, the tertiary here usually have a mixture of the alpha and the beta. So you will look something like this, and then a zigzag, and then circle again, and then zigzag. So it's like 1 is included on 2, and 2 is included on 3, and 3 is included on 4. So these are just, if you notice, it's about their bonds and folding. So let's look at the mark scheme. For primary, it's the order the sequence master which the protein is composed. We have the peptide bonds between them. And these usually control the levels of the structure. That means depending on their amino acid sequence, you will know the structures ahead. Secondary structure. This means these chains are folding, right? And they're held by what? By hydrogen bond, which usually is between the amino and the carboxyl group. You can have the alpha or the beta pleated sheets. And usually they have a hydrogen bond, so they keep the stability going on. So you already got enough of the points here. Tertiary. They are polypeptides still folding in coil, right? And they start to have a complex molecular shape. So it's a 3D shape. Here you can see that it's all about the R groups. That's what tertiary is. That includes hydrogen bond, disulfide bridges, ionic bond, hydrophilic, hydrophobic interactions. And example of tertiary structure will be the enzymes, right? So as I said, then we get to the quaternary structure. This is where you have multiple polypeptides and prosthetic groups, as I was mentioned. And these usually are inorganic, these prosthetic. And this is what I was talking about, example, the heme group or the iron. And there's a protein containing prosthetic group is usually conjugated protein. And here, the quaternary structures may held together by a variety of bonds, just like the tertiary. Now here it doesn't show, but usually they can talk about how hemoglobin is part of the quaternary structure. So here is where you're going to get most points. Usually this can be an essay of 8 points. So when it's 8 points, you just basically take 2 for each one of these guys. And what I would say is for this one, you can say that it's peptide bonds for primary 1. For primary, for secondary, mention about the hydrogen bonds, the alpha and the beta helix. That's probably 2 points. For the tertiary, you can mention about the R groups. Probably going to get points here 1, 2, and 3D shape, or even 3 points with the enzyme. And the quaternary multiple groups, you can also talk about the conjugated proteins, and you probably have hit all the 8 points. So here's the future questions that can come out of the IB exams for the topic 7.3. These questions have been answered through my lecture video of 7.3. So these haven't really been commonly, but you never know. But the ones that I showed you was the most common ones, but these could be potential questions in the future, so make sure you also know how to answer them. And that's pretty much for the section 7.3 on long answers. Thank you.